Well, good morning, everyone. It's 9 o'clock and time to start our scheduled webinar on the nitrogen cycle. We're glad all of you could join us. We'll remind you that this presentation will be recorded for later viewing. It will be posted on our Nutrient Economics website, and I'll have the web address for you at the end of the presentation. Also, this session has been approved for CCA continuing education credits. We'll supply the QR code for you at the end of the presentation and instructions on registering for those credits. So with that, let's get started on this tremendously complex subject, and we're going to try to keep things as simple as we can today. As many of you are familiar, there have been many textbooks, dissertations, theses written about this subject, so we certainly can't discuss everything about the nitrogen cycle, and we can't get into too many details in the time we have allotted, but I want to talk about some of the key concepts here. And it seems to be frozen. Apologies right now. Okay, so as we look at the nitrogen cycle and the many things that go on within it, we can say with certainty it's a dynamic system. It changes rapidly. There are a variety of different inputs and losses from the nitrogen cycle. There are different forms of nitrogen and transformations, and these forms are continually changing from one form to another. And understanding this can help us with management implications. Now, today, we're not going to discuss much in terms of management, but understanding of the nitrogen cycle can lead us to a better understanding of the source, rate, time, and place choices that we have and how to make better management decisions. So we want to discuss these items, and hopefully the principles will give you help in management. All right, so this is a visual of the nitrogen cycle. This visual is posted on our economics website, and you can go to this graphic and click on each of these items where it says more and get a little bit of detail about each of those. So I encourage you to follow up with that if you have some questions. Also, as we go along, there's a Q&A box that you can submit questions, and we'll try to get to as many of those as we can during or at the end of the presentation. So don't hesitate to send your questions in. You can also submit questions on the website. We have an Ask the Agronomist link, so you can follow up there, or you can send an email to me directly. I'll provide you my email address at the end. So there's a variety of ways you can get clarification or more detail on any of the things that we discussed today. So as we look at the nitrogen cycle, I want to separate all of these things here into some groups. So we look first at some of the main components. Atmospheric nitrogen is obviously the greatest pool of nitrogen in our environment. Almost 80% of the air we breathe is nitrogen gas. But that nitrogen isn't available to plants. It has to be converted to other forms, and there are there's processes by which that can happen. In the soil, the largest pool of nitrogen is the organic form. We'll talk a bit more about that. And then we have the ammonium and nitrate forms, and those are the two forms that are taken up by plants. Those two forms are also very reactive. So we want to know how to manage those, and those are the forms we typically apply as fertilizer as well. So then we have a variety of ways nitrogen can be added to the soil, or inputs to the nitrogen cycle, from organic materials such as manure and plant residues, fixation by legumes, atmospheric deposition, and also fertilizers. That's the business we're in, so we really want to understand the fates of these materials, of these inputs. In balance to that, we have losses. Some of these losses are have environmental, well, all of these losses have negative environmental consequences, with the exception of crop harvest. Every time we harvest a crop, we remove some nitrogen, and that's really a desirable thing. We're usually looking for that nitrogen as a source, contributor to protein, either in animals or in our own food. So crop harvest is something that's desirable. We want to get as much of our nitrogen into the crop as we can. The other losses have negative consequences, and so we want to understand those processes and how we can avoid those losses or minimize those losses. Then there are a few terms, and we'll define these terms, explain the differences between them: mineralization and immobilization, nitrification and denitrification. And these are transformations that are occurring virtually all the time in the soil. And these are biological processes, and processes over which we often have little or no control. And they're also influenced by weather, over which we have no control. So this whole system, this complicated system, is driven by many things that we can't control. We can only learn to manage around them. All right, so let's talk about these transformations and the components. So the four key transformations that are highlighted on the graphic are mineralization, immobilization, nitrification, and denitrification. There are others of, I'll say, lesser importance to our discussion. They're certainly important in the soil, but they enter less into our management discussions. So mineralization, that's simply the process of biological conversion of organic nitrogen to mineral forms. So we already said that the organic pool is the largest, but that has to be converted to plant available forms to be taken up by the plants. Immobilization is kind of the reverse of that. It's a biological conversion of those mineral forms into organic forms. So as the microbes decompose organic material, they also need a source of nitrogen, so that gets incorporated into their proteins and goes into the organic fraction. The next process we'll mention is nitrification. So this is bacterial conversion of ammonium to nitrate. It's a specific species of bacteria. There are actually two species involved. It occurs in aerated, moist soils by these oxidizing bacteria. It's in warm, moist soils. It's a fairly rapid process. We'll discuss that a bit more. And then finally, denitrification, which is the conversion of nitrate into one of several nitrogen gases. And the gases that are produced are dependent on the environmental conditions, and this occurs in anaerobic soils, soils that are oxygen deficient, as bacteria strip the oxygen from nitrate. So certain anaerobic species of bacteria look at nitrate, we could say, in a sense, as an oxygen source. And in the process, they convert that nitrate into either nitrogen gas, nitrous oxide, or nitric oxide, one of several gases. And nitrous oxide is the one we're usually most concerned about because of its environmental effect as a greenhouse gas, greenhouse forcing gas. All right, so some of the components. Let's talk about organic nitrogen, because this is so important in the nitrogen cycle. It's kind of at the middle of a lot of these processes. About 95% of the nitrogen in soils, and this obviously varies, is in the organic form. And just to do a little bit of arithmetic, 1% organic matter is going to equal roughly about 1,000 pounds of nitrogen. So there is a lot of nitrogen, even in low organic matter soils. There's a lot of nitrogen there that the plant can't use. And it needs microbial activity to convert it. And if we look at the ratio of carbon to nitrogen, phosphorus and sulfur, you can see that there. So roughly a 10-to-1 ratio of carbon to nitrogen in the soil. Now, this organic nitrogen mineralization is a real important factor in in determining our fertilizer nitrogen requirement. Unfortunately, we don't really have a good way to predict this. We make estimates, and many people have studied this at length, and it's still one of the most difficult things in determining a nitrogen rate to apply. Looks like we have a comment that's come in on the questions saying, unlike P and K, there's no reliable soil test to allow us to determine the dose of nitrogen. And that's largely true for most environments. Now, here in the Western US, where we don't have as much rainfall and we don't have as much mobility of nitrogen in the soil, a lot of us use a soil nitrate test before the crop is planted, and that's helpful because that nitrogen is available. But the participant is correct in that we don't have good soil tests for the most part to predict this availability from this organic fraction. There are some tests that have been developed that are trying to estimate that. There's lots of questions about that, and it's not clear yet, in my opinion, how well they actually predict available nitrogen, but there may be some promise there. Okay, so just to take a look at this organic pool a bit more, this is some older data from Dr. Paul, who has written a number of things on organic matter cycles, so a famous soil biochemist, and he published the data I'm showing here. You can look at kind of the breakdown of this, but really the important thing is to look at the bottom two lines of this chart, the very stable forms of organic material in the soil hold most of the nitrogen, but don't contribute very much in the short term because they take many, many years, even hundreds of years, to decompose and release that. So when you look at the contribution to that mineralized pool, it's largely from active biomass or non-biomass, fresh crop residues, fresh plant roots that have roots from plants that have died and are decomposing, or dead organisms in the soil. So that's where, when we talk about this contribution to plant available nitrogen, that's where most of that is coming from, rather than from the humus. The humus itself is a very important material in the soil, but it doesn't contribute a lot of nitrogen. So nitrogen is needed by these residue decomposing organisms, and the conversion of that nitrogen into their proteins is called immobilization. If the residue nitrogen is low, then those organisms will use soil nitrogen, and that makes that nitrogen temporarily unavailable to the crop that we're trying to grow. So just kind of generally speaking, carbon nitrogen ratios of the organic material greater than 30:1 favor immobilization. At that point, there's not enough nitrogen to balance the carbon for those microbes, and so they will tie up soil nitrogen. Carbon nitrogen ratios in the organic material of less than 20:1, so these are materials that have more nitrogen in them, they tend to favor mineralization. And somewhere in between that range, those processes are approximately equal. Now, it's important to remember that both of those processes, immobilization and mineralization, may be occurring simultaneously. Organisms are digesting organic material, they're growing and reproducing, creating proteins, and they die, and those decompose usually fairly quickly. And so those processes are continually going on, often at the same time. So when we talk about immobilization, mineralization, or maybe a better term is net immobilization or net mineralization of that nitrogen. Here I'm showing just some approximate carbon nitrogen ratios of various materials. As we said, the soil itself is approximately 10:1. We look at a legume like alfalfa, that's a higher nitrogen plant material, about 13:1, 15:1. Rotted manure, so this would be something like your compost and other similar materials, about 20:1. So again, there we would expect a net mineralization. And then as we get into various crop residues, such as corn stalks or small grains, the carbon nitrogen ratio is much higher. Those materials are very low in nitrogen, and so they're more difficult to decompose, and we expect a greater amount of immobilization. And then you can look at things like coal, shale oil, hardwood dust, spruce sawdust. So these materials are very difficult to decompose for a variety of reasons, but one of them is they're very low in nitrogen, and the organisms really need a source of nitrogen to decompose those. So you may put sawdust in a compost pile, but you need to make sure that that's balanced with some amount of nitrogen to cause that to decompose. And that's why in our compost pile, you hear rules of thumb of approximately equal materials of brown and green is one way I've heard it described. So if you're putting dead leaves into your compost and fresh green grass clippings, those would balance each other in terms of the nitrogen and carbon ratios. All right, so here's a little demonstration I saw at one time, and this is this was in an Iowa cornfield, and it was presented at a field day. And we have here the a couple of strips. One strip had received 80 pounds of fertilizer nitrogen. The strip in the front had no nitrogen. And this little block here in the lower right-hand corner had no nitrogen, but something else was added to that. And so there was a bit of a quiz session. The presenter of this demonstration was asking people what happened there. And his explanation was he had applied sugar, sucrose, to this little plot. And sucrose or sugar has no nitrogen. And so that carbon source, a readily digestible carbon source stimulated the microbes in the soil, and they consumed much of the soil nitrogen that was present. And so you see this severe reduction in growth there. Okay, there's a question here. A question and a comment. So one comment that biofertilizers are essential to supply nitrogen, but we hardly know the rate of N these will supply to the crop, and how do we compensate for that? I have to say, I do not know the answer to that question. That's an area that's receiving a great deal of study. Biofertilizers are becoming more and more popular, but I don't think it's clear. I don't think we really know how much nitrogen they're going to contribute to the crop. If we know what the material is, we can make some estimates, but I have to say that's not a question I know the answer to. I think it's something the jury's still out on that. Then the other question, does humic acid stabilize nitrogen like Instinct? I've heard some companies claiming this. Okay, so for those who may not be familiar, Instinct is a nitrification inhibitor, and we'll talk a bit more about that, but it basically, in short, it slows the conversion of ammonium to nitrate. Does humic acid do the same thing? Now, I have heard these same claims. However, I have not seen scientific evidence that humic acid behaves as a nitrification inhibitor. So again, that's a question that I think has yet to be validated in the research. Lots of claims being made, and I'd like to see those actually answered by some independent research, because I haven't seen that to this point. Maybe it does, but I haven't seen the evidence. Okay, now we talked a bit about the contribution of organic nitrogen to fertilizer, and here is some historical data for a field. And we have in this chart the yield on the left and the agronomic optimum nitrogen rate on the right. We have a check yield in the gray bars, so that's the yield with no fertilizer nitrogen. And we have the maximum yield that was produced and the agronomic optimum N rate for each given year. And you can see that in some years, there was a great response to fertilizer. For example, here in 2003, there's a large response to fertilizer. The agronomic optimum N rate is quite high, close to 250 pounds of nitrogen per acre. So there's a big response. But in another year, say year 2000, the yields are virtually the same whether you applied fertilizer or not, and the agronomic optimum is less than 50 pounds per acre. There are a couple of years like that. And so 2004 also, there's no response to fertilizer nitrogen. So how do we predict this and know how much fertilizer to use when we have such tremendous variability from year to year in the field? And that's a question that's still out there. And as we said, many people have studied this, are studying this. And one of the great mysteries of nitrogen management is how do we get a handle on what the soil is going to supply? Here's another example, a very similar kind of study, long-term study, where we look at the economic optimum N rate year to year, and some years it's zero. And so we might suppose, okay, in a really dry year, this is winter wheat in Oklahoma, so there are years when it doesn't rain very much. And in a very dry year, maybe we don't get a response to fertilizer nitrogen. Maybe we don't have the capacity to produce much yield. On the other hand, we might also have a year that is ideal for mineralization of soil nitrogen. And in that year, the soil supplies everything the crop needs. And so we don't get a response. So these are kind of the extremes that we have. But you look across here, and in some years, it benefited to apply 100 pounds of nitrogen per acre. And in other years, nothing, and lots of variability in between. So again, this is one of the great difficulties in predicting the appropriate nitrogen rate. All right, so let's talk about the other components. Ammonium nitrogen. This is a key component in terms of nitrogen management and understanding what it is. It is what we call an immobile nitrogen form. It's a exchangeable cation. It is plant available. The interesting thing about ammonium is even though it's plant available, it's not taken up as quickly as nitrate. While there are certainly advantages in the plant to some amount of ammonium, the plant can't take up as much typically in the ammonium form as it does nitrate. There are some exceptions to that. Ammonium is generally the first product of mineralization. So as those organisms are decomposing organic nitrogen, ammonium is produced first, and then it will convert to nitrate by nitrification. The residence time of ammonium in the soil is usually fairly short. So if we go sample the soil at any point in time, we'll usually find quite low levels of ammonium and much higher levels of nitrate. The exception to this would be shortly after the application of an ammonium fertilizer or an ammonium producing fertilizer. Ammonium can also be converted back into organic nitrogen by immobilization. So nitrate is a mobile form of nitrogen. And when we say mobile, that simply means it can move in the soil, and basically nitrate goes wherever the water goes. So if we have water moving through the soil, nitrate will move through the soil. If we have water evaporating from the soil surface, nitrate can move up to the soil surface. So it's highly mobile. It's not held on the cation exchange complex. It's taken up very rapidly by plants by mass flow. And it is kind of the end product of decomposition, of mineralization, of that process we call nitrification. And it is the form that plants take, from which plants take up the greatest amount of nitrogen, most plants. Now, an exception to this would be rice in a paddy system where we maintain an anaerobic environment, and rice takes up virtually all its nitrogen as ammonium, and it does very well that way. The conversion to nitrate in that system would be a bad thing because that nitrate would be lost by denitrification. Okay, so here's a chart that's often cited. This is found in many textbooks, and it simply gives the rate of nitrification, the conversion to nitrate at different temperatures. So if, for example, we're applying an ammonium form in the spring or early summer in warm soil temperatures, so here at 75 degrees, that conversion is virtually complete in about a week's time. So that conversion is quite rapid in warm soils. In cold soils, say if we apply anhydrous ammonia in the fall and the soil is going to freeze up, that ammonium can stay in that form for many weeks if the soil stays cold, because that bacterial process is much, much slower. So this is why for fall nitrogen application, we make the recommendation that that ammonium forms be applied when the soil is cold to slow that conversion to nitrate. We want to keep it in the ammonium form through the winter, and then in the spring it will start to convert to nitrate, and that helps reduce the risk of any loss from the nitrate form over winter. Okay, so let's talk about some of the inputs. So one of the inputs that we would consider is atmospheric fixation and deposition. Now, in some areas, this is more important than others, but lightning, in essence, can oxidize that nitrogen gas in the atmosphere to nitrate, and then by rainfall, that's deposited into the soil. Ammonium, which is also in the atmosphere, can also be deposited by rainfall. Now, we can access data on that deposition from an EPA program called the National Atmospheric Deposition Program. You can see a website there. You can Google this and you can come up with all kinds of data on deposition. But here's a map from 2018 showing nitrate deposition across the US and also ammonium deposition. Notice that the scales are different on these. So the highest areas in nitrate are approaching 15 to 20 pounds of N per acre as nitrate. So that could be a significant contributor to crop production that we should account for. But if we look at the Western US, there's very little nitrate deposited. So it's really of no importance agronomically. In terms of ammonium deposition, oh, and I should add, this nitrate deposition has declined dramatically over the last couple decades as a response to the Clean Air Act, which has forced manufacturing to reduce the emissions of nitrate and sulfur compounds into the atmosphere. And so if we look at the sulfur deposition map, it's very much the same. It's declined dramatically in the last 20 years to be almost insignificant in most areas. In terms of ammonium, you can see the kind of the northeast quadrant of the US has higher ammonium deposition. There are a variety of causes speculated for this. There are feeding operations in those areas. There is a fair amount of urea used as fertilizer in those areas. The causes aren't exactly clear. Some of the atmospheric chemists have proposed that it's related to the decline in nitrate and sulfur. So nitrate and sulfate in the atmosphere would combine with ammonium, and then it would precipitate out and fall to the earth, where without those acids in the atmosphere, the ammonium has a longer residence time. So some have proposed that it's related inversely to the decline in nitrate and sulfate. But anyway, these are still very small amounts, on the order of a few pounds or kilograms per hectare, that are actually deposited on the soil. So agronomically, while atmospherically and in terms of the environment, it may be very important, but agronomically, it doesn't have a lot of impact on our management decisions. All right, one significant input into soil nitrogen is what we call bacterial fixation. And this is primarily, or the fixation that's most important to us, is that occurs in nodules on the root systems of legumes. So certain bacteria, generally Rhizobia species, infect the plant roots. They form little colonies, as I've illustrated here in these little pink circles. Pink because a healthy nodule, if you cut it open, will have a little bit of a pinkish color. And these bacteria take the nitrogen gas from the air, and using energy from the plant, they convert that into various amine or amino compounds and supply those to the plant, and the plant can metabolize that. So it's what we call a symbiotic relationship. The plant supplies the energy, the bacteria supply the plant with nitrogen in return. And many of these legumes don't need additional nitrogen fertilizer, although we're finding that in some high yielding systems, it may be beneficial to add small amounts of nitrogen at certain times during the growing season. Now, we should note that there are also free living bacteria in the soil that are not associated with the higher plant that can also fix nitrogen, some blue-green algae and some other species, but those amounts are usually pretty small and not a significant contribution, although maintaining good, healthy soil biological systems can probably augment that. Okay, let's talk about animal manures and biosolids. These can supply primarily organic nitrogen and some ammonium. So keeping in mind that when we apply these materials, that organic fraction has to be mineralized to become plant available. So the challenge with managing these materials is knowing when that nitrogen is going to become available, how much is going to become available in the growing season. Some of those compounds may carry on for several years before they're completely decomposed. So we really should be testing the material to know how much nitrogen is in it. And then there are estimates of how fast that can become available in different systems. Those are really estimates. And if you're not testing the manure, we rely on book values for their nutrient content. And those are really very approximate. And the materials vary widely. So those approximations may not be particularly accurate for any given situation, but in the absence of a test, it's the best we can do. But it is difficult to know how these materials are going to decompose and how much nitrogen is going to be released. These materials can have great benefit to the soil as they add organic material, but managing nitrogen is a bit complicated. We can say much the same about crop residues. Much of the nitrogen in those crop residues is organic nitrogen. They typically don't contain high levels of nitrate or ammonium, although certain fresh residues may. One that comes to mind, a crop that I've worked with a great deal, is sugar beets, where we cut off green tops that are fairly high in nitrate nitrogen at that time. They're not a mature brown material like wheat straw or corn stalks. So there may be nitrate present. We think about vegetables being harvested and the green waste that's left in the field. Those would be higher in nitrate nitrogen. But primarily in plant residues, it's organic nitrogen that we're concerned about. And that, again, just like manures and compost and other biosolids, has to be released or mineralized by bacterial or microbial decomposition of those materials. And so all those things we said about carbon nitrogen ratios come into play. And sometimes we would even add a small amount of fertilizer to stimulate that decomposition and feed those organisms if we're adding a high CN ratio material. Okay, fertilizers. And this is the input that many of us manage. This is our business in Nutrien, and many of those on the participating today are also involved in the fertilizer industry. So this is a key point for us to understand, and understanding the management of fertilizers, which in the beginning I said we wouldn't get into too much today, but this is what all this leads to. So atmospheric nitrogen, the way we produce fertilizer is react atmospheric nitrogen with natural gas at high temperature and pressure to produce ammonia. And this is the precursor of all nitrogen fertilizers. It's a high energy process. That N2 molecule is a very stable, non-reactive molecule. So it takes a lot of energy and a catalyst to split that apart. And then using the hydrogen from the natural gas, we make ammonia. And then we have something that's both plant available and it's reactive, and we can make a lot of other things with that. This process was developed in the early 20th century by a couple German scientists, Fritz Haber and Karl Bosch, who worked together to invent this process and commercialize it, and really revolutionized the world, and a really significant contribution to global society, in that having a readily available form of nitrogen and abundant, low-cost nitrogen fertilizer has allowed the world to feed the billions of people that now live here. It's been estimated that 40 to 50% of global food production is attributable to the impacts of this process. So it's a very significant discovery. I've heard it called one of the most significant discoveries in the history of mankind because of the impact it's had on our prosperity and lifestyle and the ability to feed ourselves. And again, as I said, this ammonia that's produced is a precursor for all other ammonium fertilizers, and we can even take that ammonia and make it into nitric acid and make other things from that. But we can react that ammonia with sulfuric acid to make ammonium sulfate. We take that ammonia and react it with carbon dioxide and make urea. So we capture the carbon dioxide that's emitted from the process, run that back and react it with the ammonia to get one of our most important fertilizers. So this is not an insignificant discovery and had great benefit. So we got a question here. Is there any research on building nitrogen fixing rhizomes on corn roots being done? This is a great question. It's a question that many people have asked. There are people who have been searching for this, and there are some who have proposed that they have ways of infecting the corn plant and other plants with nitrogen fixing organisms. I've not seen anything commercial. There's been a lot of talk about it. I used to think it wouldn't happen in my lifetime. Maybe it will, because our technology is advancing so fast and our ability to modify plant genetics. Maybe something will happen, but at the present time, there's nothing that I know of in the commercial sphere, although I have seen some things presented in research. They're not really functioning, the ones that I'm familiar with that I've seen are not functioning the way rhizobia and legumes function and actually forming nodules. But there are other mechanisms being studied. So maybe something happens, but even those who've been working on this haven't proposed that they would completely be able to replace nitrogen fertilizer. So, you know, it's not like nitrogen fertilizer is going to go away anytime soon and still be able to feed the world. All right, so now let's talk about some of the losses. And these are the important things when it comes to management. And let's just say up front that this nitrogen cycle naturally is a very, what I call, leaky system. It is susceptible to losses. When we have nitrate in the soil, it's highly mobile. The plants need that nitrate, and so we walk a fine line between supplying what the plant needs and having nitrate in the soil that could be lost. And so again, this natural system has these complexities that are driven by unpredictable drivers, weather and soil conditions, and all these microbial processes. And so it's tricky to manage these losses. But if we understand how they occur and the processes involved, then that gives us insight into some things we can do. So to summarize, first, before we get into a little detail, we'll just describe what these losses are. So we talk about volatilization, or as my Canadian colleagues like to say, volatilization. It took me a few years to learn how to say that working with a Canadian company, but I think I do okay now. This is a result of often a choice of the source or the application method, and it's simply a loss of ammonia into the atmosphere. We're going to describe how that happens. Runoff or erosion. And the reason I put this slide together is to emphasize a certain point here, and you'll see when I'm finished here. Runoff and erosion are a result of excess water coupled with insufficient infiltration. If the water runs off the field, it can take nutrients with it. Now, usually, this is not a major source of nitrogen loss, because usually nitrogen will infiltrate into the soil before it moves. So as the water, as a wetting front advances, you have water moving across the soil, as that wetting front advances, it goes into the soil first until it exceeds the infiltration capacity, and it's taking that nitrogen, soluble nitrogen, with it into the soil. Now, if that water comes very quickly and we have runoff occurring very rapidly, and we have, particularly if we have erosion of topsoil and that organic fraction, yes, we can lose significant amounts of nitrogen if we're actually removing topsoil. The loss of soluble nitrogen, say nitrate, in that water is usually quite small, but there are certainly exceptions. Denitrification, a result of excess water and poor drainage. So the water fills the soil, it ponds, the soil becomes oxygen deficient. And as we explained, those bacteria look at that nitrate as a source of oxygen and convert it to gases. Leaching, also a result of excess water coupled with excess drainage. So in this case, the water is moving through the soil rather than accumulating in the soil or running off, and it carries the nitrogen down through the soil. So I built this slide to illustrate that many of our loss mechanisms are a result, of excess water coupled with some kind of soil condition, and neither of those are very much within our control. Now, if the water is a result of irrigation, we have some control over that. But we can't do a lot about the soil conditions, and we can't do a lot about precipitation. We can't do anything about precipitation. So I'm emphasizing again that these are a result of processes that are mostly out of our control. Now, one of the ways I like to think about nitrogen loss is kind of two basic types of loss. There's this short-term risk of volatilization where urea or urea-containing fertilizers are placed on the soil surface. And in the short term, it takes to convert that to ammonium. Free ammonia can accumulate. And so those losses occur within a few days after that application if the urea is not incorporated into the soil in some way and trap that ammonia. If it stays on the surface and we have conditions for that loss, we can have volatilization. But once the conversion is done, the risk of volatilization is negligible. On the other hand, there's a risk of nitrate in the soil, and we want that nitrate there because we need it to feed the crop. But anytime there's nitrate in the soil coupled with that excess water, we can have leaching or denitrification or runoff, as the case may be. And this is a season-long risk, or anytime we have nitrate sitting in the soil, we can have that loss if rainfall or irrigation events or weather conditions occur to drive that loss. So one is a short-term problem that we may be able to manage with some of our practices, and one is a more long-term problem or challenge that we have to manage season-long. All right, so let's talk about volatilization first. It's most often associated with surface-applied urea fertilizers, although not exclusively. There are conditions such as improper application of anhydrous ammonia. If the injection slot doesn't seal properly, we can lose ammonia, and that would also be called volatilization. If we have a high pH soil, ammonium sulfate could also volatilize, although that's less common. So usually we're concerned about ammonia or urea. And those losses are greatest when it sits on the surface. We have low cation exchange to absorb the ammonium. We have surface soil moisture to dissolve the urea, but not enough to move it into the soil. And we have evaporating conditions, the sun shining, a breeze is blowing, we have lots of residue. And this is driven by an enzyme called the urease enzyme, which is responsible for converting urea to ammonium. And in the process, one of the intermediates is ammonia gas. And if it's on the surface, some of that escapes. And here's just an example of a study. And here we have urea applied to dry soil and applied to mud or wet soil with and without urease inhibitors. NBPT is a urease inhibitor sold under many different brands, Agrotane being probably the most well-known. So here we have urea applied to the mud, this top line. So wet soil, it dissolves, it starts to hydrolyze, and volatilization can occur. And in this case, the losses were in excess of 30%. Then we have urea applied to a dry soil, and you see the loss tops out at about 15%. And probably because the dissolution of the urea was much slower, not enough moisture to dissolve the urea, and so the conversion is slowed. When we add the urease inhibitor, so here is the NBPT added to the urea applied to mud, we've reduced the loss substantially, cut it to about a third. And applied to dry soil, really the loss is just a few percent. So the conditions are important, and this also illustrates that there are tools we can use to help reduce those losses, inhibitors that we can apply that buy us some protection for that short period of time that is at risk. So runoff and erosion, we mentioned this a little bit. It could be a loss of any of the different forms in the soil. It again occurs when the precipitation rate exceeds infiltration. You have water moving off. And as I said, it's in the absence of topsoil loss, it's usually a fairly minor loss mechanism. Denitrification is exclusive to nitrate. So it's a specific transformation occurring from the nitrate form. It occurs when we have warm temperatures, a fresh carbon source, so something to feed the organisms, the bacteria, and we have oxygen deficient conditions or anaerobic conditions. And that's where denitrification occurs. So it's typically greatest in the spring and early summer. That's typically the time when our soils are wettest and we have more nitrate present. So as spring comes on, mineralization picks up, nitrification picks up, we start getting accumulation of nitrate. Maybe we've applied some fertilizer. So we have an accumulation of nitrate, and we have that coupled with warming soils and typically the wetter part of the year. And that's usually when denitrification is greatest. Now, my personal opinion is that denitrification is underappreciated in its severity. We talk a lot about leaching because of its impacts on water quality, and we hear a lot about that. But I've seen in the field circumstances that suggest to me that denitrification may be even a greater loss than leaching in many of these environments. Here's some examples of some things that affect denitrification. The table on the lower left, you can see different temperatures, 55 to 60 and 75 to 80, the time that the soil is saturated. So at cold temperatures, even with 10 days of saturation, our loss is 25%. Now, I don't want to minimize that. That's certainly significant. But in a warm soil, those losses are much, much greater. And in this one case, almost 100% loss of the nitrate in. So certainly these can be serious problems in say in the Midwestern US, where we have lots of poorly drained soils that accumulate water. We typically have excess rainfall in the spring and early summer. And we often have crop residues in fairly high organic matter. So certainly temperature is an issue, and also the amount of water-filled pore space. So the more extreme the oxygen deficiency, we could say, the further this process is driven towards N2 gas. Now, the one that we're most concerned about is this nitrous oxide because of its environmental impact. Okay, moving on. Let's talk about, here's also an example showing the effect of different soil types. So in the sandy loam, we have an initial surge of denitrification. Maybe there was some excess water for a short period, but that soil drains fairly quickly. The anaerobic conditions dissipate. We're in a clay soil. Those conditions persist for a longer period of time. And so typically we have greater denitrification losses in heavier soils or soils with barriers to permeability and infiltration. All right, let's talk a bit about leaching, and then we'll wrap this up, and hopefully there'll be a time for another question or two. Leaching is simply movement of nitrate below the rooting zone. Where we're really concerned is when it goes beyond the root zone into groundwater, which creates contamination of water supplies. It occurs in well-drained soils and also artificially drained soils. So when we create the movement of water through the soil with artificial drainage, and we couple that with precipitation and or irrigation exceeding the water holding capacity of the soil to the point the water moves through the soil, then it carries nitrate with it. Remember, we said nitrate is soluble, it's mobile, it goes wherever the water goes. So if that water is moving beyond the root zone, it's taking some of the nitrate with it. So here's an example of a recent study out of Minnesota, and we're simply looking at some different fertilizer treatments. But what I wanted to point out is how this is related to these rainfall events. So we go back in April here, and there's no crop growing here yet. So this was planted to corn. The blue vertical bars are precipitation events. And so we have a number of events here early in the summer that are roughly an inch, 3/4 of an inch of moisture. And every time you see, so like here in mid-April, there's several events and you see a spike in the leaching loss, so the nitrate loading. So these are measured in lysimeter plots where they're capturing all the drainage from the plot and then analyzing that. Then here in May, we have another cluster of rainfall events, another spike in the nitrate loading. Then there's this big storm in early July, about 4.5 inches of rain, and you see a big jump again in the nitrate load. And then from there on, we have lots of precipitation events, but we don't see the big increases in nitrate leaching. So what's happening in the middle of the summer? Well, one, we have a crop growing that's using a lot of water at that time. And that crop is also using all the nitrogen it can get at that time. So the crop is taking up a lot of nitrate, it's taking up a lot of water. And so downward movement through the profile is greatly reduced because the crop is consuming what it can get. So you don't see the big jumps later in the summer when we have those rainfall events. Now, once the crop is off, if we start having rainfall events again, you might see loss of nitrate if there's residual nitrate left that the crop didn't use. And so if we're over fertilizing and we leave excess behind, that becomes susceptible for loss after crop harvest. And here's another illustration, some of those same things. What I want to direct your attention to is the late summer. So we have rainfall in the black, drain flow in the red. So you see early in the year, we have a lot more drainage flow and nitrate load. And then in the later summer, we have rainfall, but we don't have drainage occurring and we don't have nitrate load. And you see the same for a second location in the lower right, the same kind of picture. So again, like denitrification, it's the spring and early summer where we typically have the greatest losses, partly because we don't have a crop that's using much nitrogen at that time. And it's also often the wettest part of the year. And one last figure on that, this is some data that's been accumulated in a regional project in the Midwestern US from many sites, different environments. But the key point I want to make here is rainfall in excess of the economic, or nitrogen in excess of the economic optimum rate contributes to greater losses. You see rates below so the economic optimum being the 0 here, and numbers to the left of that, or values to the left, are rates less than the economic optimum. Values to the right are rates higher than the economic optimum, but you see little change in leaching loss, or I should say residual soil nitrate. See a little change in nitrate level up to the economic optimum, but when we exceed that optimum fertilizer rate, now we have these large accumulations of nitrate, and that's the nitrate that's going to be susceptible to leaching if we get rainfall. All right, so harvest, this is obviously our goal. And while this is shown as a loss because it's a loss from the soil or removal from the soil, this is what we want to have happen. We want to get the nitrogen into the crop. We either want to keep it in the soil or get it into the crop. And when it goes off in the crop, it's going into food or feed for animals. That's the desirable outcome here. That's what drives the production of yield, and nitrogen efficiency is getting more of that nitrogen into the crop and less going into these other losses. So when we think about management, we think about all these different loss mechanisms and transformations. We can begin to think about, okay, certain events, weather events may be driving my losses, certain application practices, timing of application, rate of application may contribute to the potential for loss. So if I can manage the amount of nitrate in the soil, I may be able to reduce the amount susceptible to loss. So these are some things to think about as we think about management implications, which is obviously a huge discussion for another webinar. We actually did a webinar on that a few weeks ago on nitrogen losses and managing against those losses. So there's certainly a lot more to be discussed there. So to wrap this up, we'll say again that the nitrogen cycle, it's complex, it's dynamic, it's ever changing, and farmers who are attempting to manage this need lots of resources. Understanding this nitrogen cycle can certainly help, but understanding also that it's heavily influenced by things that we can't predict and don't have control over. So it's about managing risk. If we could account more accurately for mineralization, we could make better recommendations, but we have a long ways to go in that aspect and really being able to make good predictions for that. Our best practices and what we in the fertilizer industry call 4R nutrient management, that's right source at the right rate, at the right time, right place, give us the best chance of optimizing this unpredictable system, because we're trying to, in those principles, we're trying to take into account all of these processes, the transformations in the soil, the timing of all these events and the crop needs and all of those different things. That's part of the framework of 4R nutrient management. So with that, we'll stay on the line for a few minutes to give you a chance to ask a few questions.